Welcome to LabYak, where we meet some of the leading scientists in today's world. And today I'm delighted to talk to Professor Palli Thordarson from the University of New South Wales RNA Institute. Palli, welcome to LabYak.
Thank you, Steve. Pleasure to be with you here today.
One of the things that when I'm looking at where you've come from, Palli, it brought up in Iceland. Were you always someone who wanted to be a scientist?
That's a good question. I grew up on a farm in Iceland, yes, and in fact one of the most remote parts of Iceland. But I was blessed with the fact that both my brother and also my grandmother were particularly sort of keen on, they were always teaching me about nature. Where we live, we have a lot of migratory birds. So yeah, as a hobby as a kid, I was recording when you saw this particular bird first time in spring. Most of them probably coming from the UK, right? And yeah, so I had a keen interest in everything to do with nature from very early on. I don't think I saw myself necessarily possibly going into science till I went to uni. I was equally interested up to that point, maybe going to IT or possibly even economics or business or something of that kind. But yeah, it was somewhat, it was starting to crystallize, I think, in my mind by end of my high school. I was really interested in science in general, but I was torn between physics, chemistry, and geology, and also when I went to uni then to do chemistry in the end at the University of Iceland. I didn't have in mind to become an academic scientist. I just wanted to get a job in industry.
Right. And then when did it become something that you saw was an academic career then? Because you made a move that wasn't the obvious move for an Icelandic person, or maybe it is to go to Australia.
No, it absolutely wasn't, as I say. So what happened? I finished my undergrad in Iceland, and I got a job for a year at the main research institute there, but in collaboration with industry, right?
Right. Mm-hmm.
And I was still sort of thinking more of those lines. I started to really get the bug for research then, and saw myself suddenly thinking, oh, maybe I should do a PhD. And look, I can't still explain exactly why I ended up picking Australia, but it was a very complicated matrix. But I think even at that point, when I decided to go from Iceland to Australia to a PhD, I was probably still thinking that once I'm done with that, I'll probably end up in industry. Right. It wasn't until sort of halfway through my PhD that I started to think, well, this is an intriguing job to become an academic. Maybe this could fit me.
Yeah. And did you have, when you went to Australia, you ended up in Sydney?
Yes.
Was Sydney always the place, or is that
no?
Random,
look, as I said, the process of me sitting in a lab in Reykjavik or something, thinking maybe I should start to do a PhD till I ended up in Sydney was very convoluted. A lot of method of elimination. I don't want to really go there because of this and that.
Right.
I was 2/3 on the way to go to America. I already done the initial tests you have to do to try to get into PhD programs in America. But I had this notion it would be really cool also to go down under. And I was thinking equally Australia and New Zealand.
Right.
Not having a lot of luck finding anything useful. And until I spoke to my boss in Iceland, and he said, oh, I know this guy in Sydney. Why don't you write to him?
Uh-huh.
So that's actually really why I ended up in Sydney, and nothing to do with, I mean, when I found out, oh, it's Sydney, that's really interesting. One of the coolest places in that part of the world. So yeah, but what happened then, I wrote to this gentleman, Max, and 2 weeks later, I'm in the lab one morning and I got a phone call and say, you want to come over? And I couldn't say no.
No, that's
so,
great.
Yeah.
And did that pick your subject matter? I mean, were you very focused already in the direction you wanted to go?
No, well, so I see myself sort of at the very core as an organic chemist. And that was my favorite topic towards the end of my undergrad, and what I was working on in my work in Iceland for over a year with industry collaboration was sort of a bit of a hybrid of chemistry, organic chemistry, and indeed biochemistry. I've always been interested in that interface. And Max was working also somewhat in that space. He was more in a more conventional organic chemistry, but very keen on this concept of bio-mimetic chemistry. And so that's what I got into that. And moved a little bit in other directions since establishing my own career. But yeah, I'm still an organic chemist in some ways at heart.
And on that journey from those the first arrival in the lab in Sydney to where you are now, you've gone through a number of. You've said you've kept in a fairly straight way with organic chemistry, but it's taken you into some really interesting areas. Just from my reading of what you've been into.
Yes.
When you look at those areas, I mean, what you look back and are most excited to tell people about?
Yeah, I think like, when just so on that topic, when you have to write a grant application, one of the worst things you have to do a grant application, you got to write about how wonderful we are, right?
Yeah.
And I was struggling with this one day and tried to get help from someone. She suggested I should say I'm a polymath.
Okay.
Because you're right, like, I mean, reflecting, yes, I came out of organic chemistry, but I mainly worked in that sort of related area, supramolecular chemistry since then, moving into, on the side, a bit data analysis of all things, yes.
Yeah.
But the core focus the last 10, 15 years has been towards peptide materials, polymer-based materials, and now even RNA. And over that, you have this sort of umbrella of nanomedicine. And I think the red sort of thread that goes through all of that work I started sort of after I started my own career is complexity in chemistry. I'm interested in trying to both understand complex chemical systems and then how to use them. And this is kind of one reason why nanomedicine was very appealing to me. It is a very big challenge. How are you going to package up a drug molecule, whatever it is, into a delivery system? Use our understanding of how nanomaterial interact with biology and to get some function out of it. But then, 10 years later, we have the lipid nanoparticles with mRNA vaccines coming to the foray with COVID-19. And that system is a really nice example of complex chemistry, because those lipid nanoparticles in these vaccines, it's a complicated system, it's a complex system, but it's actually a lot simpler than biology. And, when people I can understand in some ways why people are a little bit afraid of mRNA vaccine technology, because it's so alien and new to them. But actually, in terms of chemical complexity, it's much less complex than conventional vaccines,
right,
which often involve, inactivated viruses and whatnot, or, you're almost doing cell extraction. In terms of how well they're chemically defined, they're actually much better chemically defined, except it's a complex mixture. It's not just, one molecule like an aspirin, but it's only really 5. It's 4 different lipids and the mRNA molecule, right?
Yeah.
But everything there plays a role, like, and in terms of making this package work, it's a beautiful example of complex chemistry, actually.
Do you think, it's really interesting you've said it like that, because, a number of people have talked about how, had COVID hit us 5 years earlier, we wouldn't have perhaps been able to do what we did. But listening to you, I'm wondering whether that's true or not, because do you think COVID actually gave us a challenge that only by 2020 did we have the know-how to do, or do you think we've had that for a while?
Yes and no. We were almost there. But the idea of using mRNA therapeutics has been floating around for 20, 30 years. Lipid nanoparticles developed in British Columbia, 20 years ago or something. But they didn't really catch on initially, partly because they kind of looked, and I will be very honest, I think the chemistry community that sort of was somewhat skeptical about it, because they kind of, this is weird. This is not a liposome, and it's not a nanoparticle, right? It's something in between. But the reason why, in some ways, mRNA vaccines then took off is that they were shown to work with siRNA, right? They solved the delivery problem there. And that sort of is starting to come through in around 2013, 14, 15, which is probably why investors had faith in companies like BioNTech and Moderna, who start their journey sort of in that period. But that all said, so yes, maybe we could have done it a couple of years earlier, but that all said, I've also heard people, in fact, from Moderna saying that when it comes to the challenges of scaling up all those technologies,
Yes. Yeah.
Even though, the principles were probably there by 2015, it could have been a big push even under, sort of a global pandemic situation to get as quickly as we did in 2020 from, But I think I wouldn't, I mean, if I remember correctly, Moderna was already in phase 1 with Zika virus vaccines in 2017. So they just, lost out because there were better vaccines developed quicker, right?
Yeah.
So,
Yeah.
But 10 years ago, we wouldn't have stood a chance, I think.
Right. That's interesting, isn't it? And if you look at where you were 10 years ago and some of the things that you were looking at, is it hindsight's a wonderful thing sometimes to plot the path to where we are today. But I mean, the nanochemistry and what you've been looking at has taken you into areas where you looked at photovoltaic cells, for instance, and how you might improve them. You've looked at some of the rules of life and how might we even have arrived where we are, looking at self-assembly. When you look at all of the things that you've been looking at, now you're the RNA Institute, is it an obvious pathway that takes you to there, or were there sort of 2 or 3 other ways you could have ended up going?
No, the RNA one in some ways is more obvious than others, and I have to actually go all the way back to my PhD there, which I accidentally ended up doing in Sydney, because the topic of my PhD was somewhere on self-replicating molecule, which then relates to trying to understand the origin of life.
Yeah.
And although for a long time that was an area which is almost impossible to get any funding in, I was always interested in doing something in that space.
Right.
And my work in nanomedicine went hand in hand with, and glad you picked this up, we were trying to make sort of what we call protocells, so making self-assembled polymer particles as a vehicle to then test out simple chemical concepts and then try to make them more and more complex. This is this whole idea that, you start off basically with a single molecule and then you start to mix things together, and the ultimate complex chemistry mixture we have is the living cell, right?
Right.
So there's a continuum there. And in a way, yes, I wanted to always to get into RNA and RNA chemistry because it is probably the key to understanding origin of life. So in some ways, yes, that all fits nicely together. I think what we were naive about 10 years ago is it was probably the biology of the body more than anything else. I was playing with both, as I say, polymer nanoparticle, and more importantly, I was very interested in trying to use peptide material, gel-like material for drug delivery. And you need someone who understands, say, cancer and biology to pull you up and say, well, actually, that's not really going to work.
Right.
Because metastatic cancers are the main problem, right? And it's just all this, I'm realizing that it's one thing trying to deliver a drug or RNA or whatever it is. It's a completely different scenario to deliver it to the right place at the right time in the body.
Yeah.
That's the main problem. And that was what the lipid nanoparticles solved in the case of the vaccines. Whereas unfortunately, a lot of the stuff we've been trying in nanomedicine still hasn't worked. And I'm working with people here in the Children's Cancer Institute. We've been trying to help them for many years, and so have many other people, come up with better ways of delivering cancer drugs or RNA drugs or whatever you want to the brain. It's really, really hard, right?
Yeah.
And so even if we understand the complex chemistry and the sort of nanotechnology here to a large degree, there is another level when it comes to multicellular organisms, tissues and cell types and all that, which we haven't quite cracked. We're getting there. That's the next one, right?
And that's great, because talking about the next one, you sort of look at even in the pandemic, there was a recognition that companies like Moderna had set themselves up as the popular phrase of cancer vaccines. And that had perhaps not gone as fast as certainly Moderna had hoped it would do. But then in the pandemic, people started to go, well, look, if you can do this with pandemic vaccines, how far are we off cancer vaccines? And I'm interested where you see the next 5 years. You know, if you think of the developments, hopefully we don't have another health emergency that comes at us like a pandemic, but we still are dealing with what is effectively a health emergency of every human, you know, whatever the percentage chances we'll all get cancer at some point.
Yeah.
Do you see mRNA as one of the RNA, sorry, as one of the ways in which we can address that?
Absolutely. And actually both types of RNA. So mRNA is a protein replacement, and particularly in sort of maybe CAR-T type approaches to cancer. And then also silencing RNA. I mean, silencing RNA, if you can get it to the right cell type, is really, really useful.
Yeah.
Therein lies the rub. Now, pre-pandemic, the problem was, yes, Moderna was actually founded to do cancer therapeutics, not to do vaccines. They just went in that direction, because the problem is still that it's very hard to deliver these drugs, whether it's RNA drugs or any other cancer drugs, specifically to a specific cancer or on cancerous cells. There's a twofold problem, because some organs are very hard to hit, like say pancreas, but then you can also, once the cancer reaches a certain state, you got all these metastatic cells going around your body. You kind of have to, so you have to both, it's sort of like you have to both have a targeted approach and then you have to carpet bomb, right?
Yeah.
And the problem with all these delivery systems, including the LNPs, is that they can't do that. They can only target specific tissues. Now, I think pre-2020, the additional issue was that there was not still as good understanding as we have now, and not to mention resources to try to do this.
Yeah.
That will be the big difference post-COVID-19 vaccines, is that, first of all, we have probably learned now even more about both the limitations and capabilities of these delivery systems we have now. But more importantly, like I think, because now it's been demonstrated to work in the context of the vaccines, it's much easier to get people on board to try to then move on to harder targets.
Yeah.
And is this not just an issue of money, because it's often just an issue of people wanting to spend their time on this, right? I mean, in some ways, I find the best thing about, say, the UNSWRNA Institute is that, yes, we got some good initial funding from the university, but everyone is enthusiastic now to work on this topic.
Right. Yeah.
Like our biggest brains are in wanting to be involved.
Yeah.
If you 4 years ago went to someone and said, hey, can you try to help me develop a nanoparticle and deliver something to the brain? But I'm busy doing this other thing, right?
Yeah.
So I think what will happen is that we will start to see some breakthroughs where we can start to deliver these RNA therapeutics to tissue types or cell types we haven't been able to do. But the other one I hadn't appreciated till recently is that at the same time, there's been a quiet revolution in bioinformatics, in genomics, and methods to measure RNA levels in the body, right? By various methods. And the people who are developing the therapeutics, they're saying, if we have all of that information, right?
Yeah.
Then we can actually design these drugs much better.
Yes. Yeah.
And that's the other thing, that, and even AI and whatnot comes into this, that you can start to think about overcoming some of the challenges with RNA deliveries, possibly by simply designing your RNA slightly better. And it's possible you could target even cell types that way.
Yeah.
On the back of our explosion in knowledge on bioinformatics and genomics and all that sort of stuff, right?
Yeah. I mean, you paint what if you could take a very positive view of the opportunities in the next 5 years in the best brains, the enthusiasm, but the knowledge also exploding at the same time, bringing that lot together. And basically, it sounds, listening to you, as if I would be quite hopeful that there will be some quite good steps forward in this area.
But there will also be failures, and or we will probably, as I say, we will, like with cancer, we've been trying to do something about cancer very aggressively for 50 years.
Yeah.
And lab expertise have improved, and certain cancers are now reasonably treatable, but then others are not, right?
Yeah. Yeah.
With the RNA therapeutics, we'll probably try to go for the ones that are really most difficult now, but maybe there are some biological reasons why we will still struggle with some of them, you know.
Yeah.
You know what I mean? Like, we can't solve every problem immediately, but I'm reasonably optimistic. And but then further down the line, what I'm interested in is when we can start to develop therapeutics that modulate RNA protein interactions in the cell.
Right.
And
in the body. Yeah.
Yeah, in particular, the interactions of so-called non-coding RNA with proteins.
Yeah.
This is probably another level of sort of control over both cellular development and whatnot in the body that we're only just starting to understand. But might underpin approaches to treating various diseases and conditions, right? So if we can start to crack that problem, then we can also maybe develop therapeutics based on either RNA chemistry or some more, possibly something else, to modulate these. But there are people arguing that this is the key to both understanding cellular development and differentiation, and possibly also the key to understanding a lot of neurological conditions, both positive and negative. So that's what I would want to be seeing us in like 10 to 20 years time, right?
That's fantastic. Yeah. And that enthusiasm that you've got for this area, how do you keep it up? I mean, do you obviously live in Australia, so what do you do in your spare time, Palli?
My spare time, probably what's keeping me most busiest in my spare time is my family, and I got 2 kids, the 12 and 10-year-old. So, and between those and my wife and I, like it's a busy household, right? So when I have a moment, I'm busy with them, and that's probably the hardest thing for me to balance is that. But I also enjoy my time with the family, and particularly seeing how the kids are growing up and whatnot. But yeah, that's probably what keeps me most occupied.
Fantastic.
When I got a spare moment. I haven't had a lot of time the last few years for sort of hobbies. You don't get a very good look from the family if you try to go away for 2 hours to do something.
No, your hobbies become your children, I think, at times like this.
Yeah, for a while, but I mean, I think we will get through that phase soon. And I do enjoy travel and meeting people too. Yeah, but I'm very excited about what's going on in this space. And now that sort of everything is open again too, things are coming back to normal in terms of how we interact with each other, both locally and globally.
Yeah, and in no small way due to the use of RNA and mRNA in terms of getting us all to a place where we're all more confident, isn't it?
Yeah. Not that the COVID has gone away in any way and shape, and I probably had it a few weeks ago for the second time, and it's still a very serious health risk, but we're at this stage where I think society has worked out we can balancing out, we can manage it. We have to accept the consequences for a while, but I have hope that one way that the virus might mutate and become even less harmful, but also that the next generation of mRNA vaccines that are coming, for those who want to use them, those who don't want to use them, they want to use them, what can we do? But those who want to use them, they will give slightly better protection. And there's also here, for instance, locally, there's a group working on siRNA antivirals for COVID as a sort of a last line of defense, particularly people who might get very sick or immunocompromised. So yeah, we might be able to manage COVID even better over the next few years, even if it's probably here to stay with us.
Well, that's a really positive note to finish on. Thanks, Palli, for your time. Thanks for joining us on LabYak. It's been a real pleasure talking to you.
Thank you, Steve. I really enjoyed that.